Our first speaker is Dr. Sarah Heilbronner, and she will be talking about understanding how and why we should study the wiring diagram of the brain. I'll turn it over to her.
All right, thank you very much for taking the time to come here and learn a little bit about neuroscience, which is, of course, my favorite topic in the whole world. So how and why should we study the wiring diagram of the brain? I'm an assistant professor here at the University of Minnesota. I've been here for about 4 years, and you all already had time to review that. So let's get right into it. So probably most of you know that cells in the brain are special. They're not like plant cells. They're not like skin cells. They have a special purpose, and that purpose is to communicate. And so how do they communicate? Well, here you've got your major parts of the cell body up here, the axon here. Are you guys seeing that? No. I'll use a laser pointer. No, I don't. Cell body here, axon here, dendrites here. How do cells communicate? Well, it's almost like having a conversation. It's the job of the cell body to think of what to say. It's the job of the axon to talk, and it's the job of the dendrites to listen. And in order for one axon to talk to some dendrites, the axon has to be in close physical proximity to the dendrites, which means that, and this is an important point, they're specialized for communication of information, but they aren't all set up to communicate with each other. Not every neuron, which is what these cells in the brain are called, can talk to all other neurons. And so which neurons are actually structurally set up to talk to other neurons is what we call the wiring diagram of the brain. So you could think of a wiring diagram setting up, some kind of piece of electronics or something, but we also use this as a metaphor for how we think about axons wiring up and talking to dendrites in the brain. And what the less colloquial term, the more formal term for that is called anatomical connectivity. It's the connectivity, what allows neurons to talk to each other in the brain. All right, so I'm interested in anatomical connectivity. Who could possibly care about that? You're going to talk to neuroscientists here today who study the coolest behaviors, the coolest thought processes, and what I'm talking about here is like the brain's blueprint. And maybe to you that sounds really boring, but I promise that it has a lot of useful features to it, and I'll walk you through a couple of ways that it's useful. So the question that I ask is, how can we use this, meaning the wiring diagram of the brain, to study and help those with brain disorders? And I had a professor once who told me that every scientific paper is a story of a bunch of people and what they decided to do with their lives. And this is no exception. So we had a particular problem. When I was a postdoc at the University of Rochester, which is in upstate New York, almost as cold as Minnesota, double the amount of snow, that's right. I was working with a graduate student from the University of Puerto Rico, very scared of the snow, who was interested in trying to figure out the nature of a particular brain problem that we see in post-traumatic stress disorder. Now, the details of this are not super important to get, but basically people with post-traumatic stress disorder have less activity in a part of their brain called the dorsal anterior cingulate cortex. It's right sort of here in the brain. Doesn't matter for our purposes. The point is, it's one particular part of the brain that doesn't respond enough in people with post-traumatic stress disorder. And what he wanted to do was go to a rodent model, specifically a rat, and see what would happen if he turned on and off neurons in the dorsal anterior cingulate cortex and its connections, and see whether he could cure a rodent model of post-traumatic stress disorder. Now, the problem was, we sat there and we thought, cool, let's go in and let's change the dorsal anterior cingulate cortex of the rodent. Wait, where is that? So we got out a rodent brain and a human brain. Well, we got out pictures of rodent brains and slices of rodent brains and human brains. No humans brains were taken out of unwilling participants' heads for this. And we said, okay, where is the dorsal anterior cingulate cortex in the human, and where is it in the rat? And the answer was that we really didn't know. And so this is a huge problem because rodents, and you're going to hear from multiple people today, I think, who probably use rodent models. Rodent studies are essential for doing translational neuroscience, which means for understanding basic mechanisms that then we can bridge to clinical practice. Because there are just a bunch of genetic and molecular and electrophysiological, there's a bunch of cool tools and manipulations that we can do in rodents that we just can't do in humans, but will eventually have impact on human disorders. And so that's great, but rodent brains are not the same as human brains. They're not just not the same size, they just don't look the same. You know how your brain is all wrinkly, or hopefully it's all wrinkly? Rodent brains just don't have nearly as many of those wrinkles. Rodent brains look like gross tiny balloons. And so how would you figure out which tiny little part of the rodent brain is the same or not as which tiny little part of the human brain? And that's the question that we wanted to know the answer to. So we figured out that we could use the wiring diagram of the brain to answer this question. So we analyzed anatomical connectivity, those axons projecting, of a particular part of the brain that the dorsal anterior cingulate cortex is part of the prefrontal cortex. It's the front of your brain. It's the part of your brain that's most enlarged relative to other species. And it's a part of the brain that might be really, really hard to model in rodents because we think it's important for things like decision making and self-control and abstract reasoning and all those things that people tell you that as teenagers you're really bad at. But I swear you're getting better really fast. And you're already better than a rodent. So feel proud. So in order to do this, we had to compare rats to monkeys, but monkeys can be bridged to humans, and rats can be bridged to mice, so we sort of have a large set of data to work with. And this is what anatomical connectivity looks like in various representations. So these are cells that were labeled after you inject dye into one part of the brain and it travels to the connecting part of the brain. So it sort of hitches a ride along the axons that are nearby. This is what neurons look like. Look at all those cool dendrites. This is with one kind of microscopy. A different kind of microscopy, you can see the boundary between white matter here, which is where all your axons are, and gray matter here, which is where all your cell bodies are. This is not a microscopy picture. This is how we start to render things in 3D so that we can see a whole brain picture of anatomical connectivity. So what we did was to use this to try to create a roadmap for cross-species neuroscience. So we created a list of rodent brain regions and a list of, in this case, non-human primate, but it could have also been human brain regions, and we made these one-to-one correspondences. But of course, there were a lot of situations where we couldn't make the one-to-one correspondence, and that is something that we were also able to figure out by using the wiring diagram of the brain. The next problem, we get all the way to humans. So, problem number 2. Can we cure brain disorders by changing anatomical connectivity? I'm betting Dr. Opitz has a lot to say about this, and you'll hear from him later. But there are a number of treatments for brain disorders, whether neurological or psychological, that rely on changing anatomical connectivity. And I want to highlight one of those, which is deep brain stimulation. So deep brain stimulation was developed to treat movement disorders like Parkinson's disease. You can put an electrode in somebody's brain, turn it on, and I'm not going to say cure. I shouldn't say cure. Strongly alleviate the symptoms of Parkinson's disease. And this is a relatively, I wouldn't say super routine because it's brain surgery, right? But a relatively routine set of surgeries that we do here at the University of Minnesota. Not me, I'm not a surgeon. Don't come to my house expecting me to drill into your brain. There are wonderful neurosurgeons and neurologists here and at many other clinics around the country who do this for Parkinson's disease. Increasingly, we're interested in doing this for psychiatric disorders in situations where it's the last resort, where somebody has failed to respond to many, many medications and talk therapies and other kinds of neuromodulatory treatments. And in fact, we worked alongside a team that looked at how anatomical connectivity in the human brain can help predict and explain where you should put a stimulator for major depressive disorder. So the only people that will qualify to do this study, and it's a much smaller number than in Parkinson's disease, are people whose lives have been incapacitated by depression for years. But when you go in and you stimulate at the confluence of these three different sets of wires, these three different sets of axons, so this is in my monkey study and this was in a similar human study, you actually seem to be able to alleviate the depression. And they're able to see it even within the operating room. As soon as they turn the stimulator on, people describe that it's like there's a weight that has been lifted from their shoulders. And so we're able to use that wiring diagram of the brain, that blueprint, to go all the way from, say, from tiny little chunks of the brain in rodents, to monkeys and to humans, to different kinds of brain disorders, and maybe even treatments at some point. And so with that, I will take your questions, whether about this or about neuroscience in general. When I was a freshman in college, I went to a fair that had a bunch of research opportunities. And so professors would get up and give a 5-minute spiel about their research. And the professor that I wound up working for all 4 years of college did that. And I thought, oh, that sounds very, very interesting. And it was just like a light bulb turned on in my head. But until that point, I hadn't thought that science in general was something that I was terribly interested in. I mean, science was okay, but I guess in my mind, there was a really big difference between studying science in a classroom and what wound up being the actual process of doing science in a lab and discovering new things yourself. And so science in a classroom was fine, but it had never super interested me, whereas once I went into a lab and heard about what it was like to make discoveries, that was that changed everything. And so I guess the advice there is there are all these really boring things that happen when you get to college that you might think are not going to have an impact. Things like research fairs and club activities, fairs and things like that. At the beginning, just go to all of them. Don't sleep for the first month or no, you should sleep, but sleep is important. Okay, yeah.
So can you talk near the microphone? Otherwise people attending.
Oh, I'm so sorry.
To hear you.
Oh, okay.
And if you have a question, in order for them to hear the questions, we need you to speak into the microphone as well. Okay. Thank you very much.
All right, another question?
How do you confine the stimulation to certain connections, if it makes sense?
Yeah, because connections are topographically organized, right? So imagine that for some reason you wanted to block the intersection of 35W and 94, right? There's places, or at least one place I know of, where those come together, right? And then places where they spread apart. And so there's one place where these three sets of axons come together, and then they spread apart again. And so if it's important to hit that one place, then you just target surgically that one location. Yeah. Right.
Does this neuromodulation treatment only work for Parkinson's and depression, or does it work for any other type of psychiatric
There's a lot
disorder?
of different types of brain disorders that it's under investigation for, and they have different, different status in different countries, and the FDA has approved some devices for some things, and other treatments for others, and so on. I was part of a big team that looked at obsessive compulsive disorder treatment. There is OCD, chronic pain, and movement disorders and depression are sort of the most common space in the United States, at least. But there are others. There are people that are looking to see whether it could help with things like Alzheimer's disease, generalized anxiety disorder. These are difficult studies to do because you need a strong scientific foundation to justify intracranial surgery, right? And you don't really, if it doesn't work, then you've you've taken on a big risk as a patient. And so, right, these are not done willy-nilly, let's say. Yeah.
Okay, we have time for one more question.
All right, so when you're talking about PTSD, you mentioned the dorsal anterior. What is that responsible for in your brain?
Yeah, that's a good question. So we wish we knew. It's probably responsible for monitoring the state of the environment and changing your behavior in response to relatively quick updates to what's happening around you. And so one theory is that people with post-traumatic stress disorder are living not enough in the moment. Does that kind of make sense? And not responding, not updating, that things are still not dangerous. Does that? But these kinds of things are very, very hard to formalize, and there are about 100 scientists that would be very angry at what I just said, right? So, okay. But 100 that would love it, so it's fine.
Great. Well, let's hear it for Dr. Heilbronner.
Okay, thank you.